All right, so today we'll start with category 3A, which is nervous and endocrine systems. I don't think we'll get to endocrine because nervous itself is like already a big chunk of material to get through. So right now we're talking about anatomy, right? We're kind of transitioning more to like an anatomical details and stuff. So a lot of the stuff this is going to be, an exposure to it, but you're not going to learn it just by me talking to you about it, right? It'll be a lecture, you'll be able to be exposed to it, but then after that, you have to, go to your Anki cards, go to your practice problems, and really internalize what we've talked about. But this is, a chance for you to be exposed to it and ask questions if you don't understand it the first time, right? So yeah, so we'll dive into the nervous system, right? And so, what is the nervous system? And the nervous system is basically divided into two sections, right? A central and peripheral nervous system. So what do we mean by that? Okay, so overall, we have a nervous system, right? And we can split it into two overarching sections, right? For now, don't worry about like all the down parts of the flowchart, just look at the top. The top, we have the central nervous system, or the CNS, and we have the peripheral nervous system, or the PNS, right? So the central nervous system is essentially central, right? The brain, the spinal cord. Those are the two components of the central nervous system. And their role is to integrate all of the data that it gets from the rest of the nervous system, or the PNS, right? And the rest of the nervous system that's the definition of the PNS. Any tissue, any neural tissue that is not part of the brain or spinal cord is considered peripheral nervous system, right? So center and peripheral. Those are the two divisions of our nervous system. So um, let's talk a little bit more about the central nervous system, right? So the central nervous system, obviously we have our brain, right? And our brain is divided into several different lobes. One thing to understand is that like we have these sections, right? You see these little blue sections, the frontal lobe, parietal lobe, occipital lobe, temporal lobe. These are all part of what's called the cerebral cortex, right? This is the outer part. And the outer part is the most, well, I guess you could say it's the most important part because it has a lot of our higher function processing, right? So the higher function processing that we have in the frontal lobe, you'll see it's like the largest part, right? Because it's all about executive function, voluntary movement, motor movement, right? And produces speech. This is where a lot of our thinking and planning happens. Then we have the parietal lobe up here, which is spatial processing, like understanding, space, proprioception, and somatosensation. So these are kind of like how we uh, maintain ourselves in the world, how we kind of understand our balance in the world and our movement in the world. Those functions are relegated to the parietal lobe. Then the occipital lobe we have in the back, which handles our visual processing, and the temporal lobe we have on the sides, which is all about learning and memory and speech perception, auditory perception, right? So these are some of the lobes that we have in the cerebral cortex. And the cerebral cortex is filled with neurons, right? And we're going to dive deep into what neurons are. Then we have the, let me see. Yeah, this is all, yeah. So just again, another explanation, frontal lobe with executive functions, temporal lobe with learning and memory, occipital lobe for visual, parietal lobe for proprioception and somatosensation, and we also have a primary somatosensory cortex at the leftmost part of this. So in these lobes, there are some spots that are interesting, that they have like very specific function. So for example, here we have Wernicke's area. Wernicke's area, there's also another area. Let me just make sure I'm not confusing them. Wernicke's area, yes. So Wernicke's area and Broca's area. There's two different areas, right? The Wernicke's area involves language comprehension, right? And Broca's area involves language production. So these are two interesting things because if one part of the brain, if this one area happens to be, destroyed, then you can lose an element of your speech, right? So if both of them are, so for example, right, if both of them were destroyed, then you could neither understand nor produce language, right? But if only one of them was destroyed, your symptom would be different, right? If Wernicke's area was destroyed versus if Broca's area was destroyed, you'd have different symptoms. Since Wernicke's area is in charge of language comprehension, if this part was destroyed, that means that you wouldn't be able to understand speech being told to you, but you would be able to produce speech since your Broca's area was intact, right? But even though you're producing speech, it would be nonsensical because you don't understand language. And that would be called Wernicke's aphasia. Aphasia meaning that the aphasia means that you're lacking that capability. So then you could also have you could have the if the other Broca's area was destroyed, then you could understand language, but you wouldn't be able to speak and you wouldn't be able to produce language. So our brain is like very specialized in that sense. Like there are spots that are very specialized and have very unique function. So we also, that specialization also carries across the left side of the brain and the right side of the brain. So the left side of the brain, and it's also, what's the word? Contralateral, yes. So let me see. Yes. So what that means is anything that if you are lifting your right hand, the left side of your brain is doing that. And the reason is because we have something called the corpus callosum that divides the brain into left and right. And the left side of the brain processes sensory information on the right side. Yeah. So the left side of the brain is sensory, primary, spatial. Yeah, okay. So the right hemisphere of the brain, used particularly in spatial awareness, emotional intelligence, intuition, and holistic thinking, right? Whereas the left side of the brain, particularly for math and science problems, logical reasoning, and analytical thinking. So at every stage, there's a lot of different divisions to the brain. And the corpus callosum is the bridge between the left and the right. And that's what allows the cross transfer of information. So now we talked about the CNS, and we'll talk about the spine in just a moment. Let me see. Okay, so we talked about the CNS here, and then we have the peripheral nervous system, right? So the peripheral nervous system is subdivided into a lot of different options because it is all of the neural tissue outside of the CNS, right? So there's a lot of different types of tissue outside of the CNS. That's all neural tissue. What the first division is between sensory and motor, right? So these are not, this is just kind of for you to understand function wise the difference, right? So when we say sensory and motor, what do we mean? So let me see, sensory. Maybe it was a little bit. Okay, here. So we have these neurons, right? We have neurons all over our body, right? But they do different things. So for example, let's say that, the classic example of like you touch something hot, right? The neurons in your fingers are going to send a signal, right? So how does it work? Well, what happens is that, let's say you've touched something hot here, right? Touch some fire here, and a signal is going to travel all the way until up your arm or whatever until it reaches your spinal cord, right? And this signal in red, right, is going to travel upwards to your brain. This is called an afferent signal, or an afferent neuron, a signal traveling on an afferent neuron. Afferent, right? Now, once the signal reaches the brain up here, right? This is a terrible looking brain, but once the signal reaches the brain up here, the brain will make a decision, right? And then send out another signal. We'll send out another signal down the spinal cord, right? Which arrives here at the spinal cord and then is shuttled off to the muscle for the muscle to perform an action, right? And this is called an efferent neuron. So what is the difference? Afferent is receiving signals and sending it to the brain. Efferent is producing signals and sending it to the muscles, right? So how do you remember this? I'd like to remember as afferent signals arrive, right? Afferent is for arrive. Oops. Afferent signals arrive to the CNS, whereas efferent signals exit the CNS. So arrive and exit. Okay, so these are the two. So this is to explain. This is to explain this, right? So the sensory division, which is the afferent division, is basically the nerve fibers that transmit signals to the CNS, right? And once they arrive at the CNS, then the CNS can send signals back down to the motor, to the efferent. They can send efferent signals down to muscles, right? Now, this itself actually has two different categories. This efferent division has two different categories. We have the somatic nervous system and the autonomic nervous system. And that's because we have two different, well, not two different, but two major types of muscles, right? We have some muscles that are voluntary, like your limbs, right? You can move around, but there are some muscles in your body right now that are working without you having to do any conscious movement, right? So that there's a big difference there, right? So the somatic nervous system is for voluntary motor nerve fibers, right? For they receive signals from the CNS, right? But there's also involuntary muscles that are getting signals. So that's a big separation. Somatic nervous system and autonomic nervous system. So the peripheral nervous system can be divided into autonomic and somatic nervous systems. The autonomic nervous system is responsible for regulating the involuntary activities of the body, such as heartbeat, breathing, digestion, and internal body temperature. The autonomic nervous system works closely with other body systems, including the gastrointestinal system and the endocrine system, right? That makes sense. Yes. And then the somatic nervous system is voluntary movement, skeletal muscle contraction, right? However, this is not the only division, right? So if we look, so simply, we have the nervous system split into a central and peripheral, right? Then the peripheral can split into somatic and autonomic, somatic being voluntary, automatic being involuntary, automatic. Now, automatic can also has two different functions. They have sympathetic and parasympathetic nervous system. And so this is essentially this is essentially like kind of a linked into the endocrine system, right? The parasympathetic nervous system is called rest and digest, right? It's a way you can think of it, because it is how everything is functioning when you want to be when you're resting, right? When your body is in a state of rest, the parasympathetic nervous system is taking over. But when your body is in the state of fight or flight, then the sympathetic nervous system is working, right? And and we're going to see these two nervous systems, what kind of effects they have on the body. So when we have the sympathetic right here, yeah. So if you need to fight or flight, right, then they're basically directly opposite, because if you're resting and digesting, your body and your organs should be doing the opposite of what you're doing if you're fighting or running, right? So for example, if you're in a fight, right, you need to see better, your pupils will be dilated. Not so when you're resting, right? Also, your heartbeat, right? If you're resting, your heartbeat should be slow. If you're in a stressful situation, your heartbeat should be fast, right? And so on and so forth. It's very common sense, right? Like you just think about like the last time that you were stressed out, right? And how you felt, and those things all make sense. Like you were breathing heavier, you were sweating, those things, right? So that's the difference. So your autonomic nervous system, right, has the ability to be influenced by hormones and say like, oh, okay, we're in this state. All of the organs should now do this, and they will signal all of the organs to do certain things, and then you have a unified uh, response to the situation. So that's basically the division of the autonomic nervous system. The sympathetic division that encourages the whole body to fight or flight, and the parasympathetic division, which encourages the body to rest and digest. Yeah. The action of the sympathetic nervous system triggers the release of cortisol and epinephrine from the adrenal glands. And these hormones and neurotransmitters work together to keep the body alert and prepared to face a stressor, such by inhibiting digestion, releasing stress hormones, and increasing your heart rate. So notice that like it's not just that they activate things, they also deactivate things, right? This is important because you'll see that the fight or flight, the sympathetic, inhibits intestinal activity. Why do we want to do that? Because we don't want to waste energy. If it's a life or death situation, all of our energy should be focused into us surviving, right? So why do we need to spend energy on digesting food when we might not live to actually, use that food, right? We need to focus on surviving, and then later when we can rest, then our bodies will go back to stimulating our intestinal activity. So it's pretty straightforward how it controls our body. So now microanatomy, right? Anatomy of a neuron. So a neurons it's a cell, right? But it's a very distinct looking cell, right? We do have a cell body called a soma. It does have a nucleus. It does have organelles. It is a cell. It just is very distinct looking. So in the cell, right, we have these little outgrowths called dendrites, and the dendrites receive signals from other axons, right? Once it receives signals, so let me just draw here. So once, oh, why can't I? Oh, I have to draw, I see. So once signals come in, right? Once signals come in, and the cell reaches a threshold, right? We're going to talk a little bit more about that threshold. Once the signals come in and the cell reaches a threshold, then the signal is going to be allowed to propagate down this, what we call an axon. This whole thing over here is what we call an axon. Well, maybe just like this, right? An axon. So it's going to go, so the signal is going to go down the axon, and the axon is basically how the signal is allowed to travel, right? To other cells, right? Because over here, there'll be another cell with its dendrites. There'll be, I'm just going to draw a crappy cell. There'll be another cell over here with its dendrites, right? So, and we're going to talk more in depth about the action potential. But essentially, there's some unique features here that that help the signals, the electrical signals to go down. Okay, so there's a helper cells. Oh, I guess you talk about action potential down here. The helper cells. And before I talk about the helper cells, I really want to talk about the action potential. So yeah, because I feel like it'll be So the action potential, right, is coming here, right? Before we talk about the actual like concentration and stuff of the action potential, let's just talk about it happening, right? So there is an electrical signal. The cell receives the electrical signal. Once it has enough electrical signal, it says, okay, this is important. I'm going to pass it down to another cell. The electrical signal is going to come, and this axon is what we call myelinated. There is some kind of wrapping around this called myelin. So what is myelin? So myelin is basically an insulation of the axon. And what it does is it actually increases the speed of the action potential traveling down the axon, right? So if we look at an unmyelinated neuron, right, the action potential is just traveling down straight. However, in a myelinated neuron, instead of traveling down straight, it actually jumps. It actually jumps because the insulated parts, you can see over here, we have some voltage gated channels, and that's what's allowing the action potential to happen. But at the myelinated parts, there are no voltage gated channels. So the signal is forced to jump, 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 jump. It's almost like the signal is skipping, right? You can think of it as the signal just walking down the axon versus the signal skipping and hopping, right? So when the signal hops, it goes down faster. It goes down faster. So myelin, right, is this insulation, and it's produced by, so we could talk about, they were saying that there's helper cells, right, that assist in maintaining the structure of neurons. So there's the myelin itself is produced by two different types of cells. One is the oligodendrocyte, and the oligodendrocyte is only found in the brain and the spinal cord. They're the CNS. Whereas in the PNS, in the peripheral nervous system, they're called Schwann cells. And these guys both produce myelin, but they're just a little bit different. So the difference is, right, we have a picture here. You can see on the left, we have the central nervous system, right? This in purple is the oligodendrocyte. And you can see that it's wrapping around a neuron and creating those myelin insulation areas, right? Now, in the peripheral nervous system, we have the same thing, but the Schwann cell is actually, look at how like the myelin is actually like embedded with the Schwann cell, right? You see the nucleus of the Schwann cell itself, right? So there's actually 1, 2, 3, 4 Schwann cells here. Whereas in the central nervous system, there's just one oligodendrocyte doing multiple myelin sheets, right? So that's already a big difference, right? Schwann cells form individual myelin sheets, but oligodendrocytes form multiple myelin sheets, okay? And obviously the big difference is that Schwann cells are found in the PNS while oligodendrocytes are in the CNS. And finally, Schwann cells can actually help regenerate neurons, right, in the PNS, but oligodendrocytes cannot regenerate brain neurons. So that's already like an interesting difference between the CNS and the PNS. And so when this myelin sheet wraps around, right, in between the myelin sheets, there's a small little gap. There's a small little gap left, and that little gap is what allows for the jumping, right? You can see at the little gaps, and these gaps have a name. They're called nodes of Ranvier. And these little gaps are where the voltage-gated channels are present, so the action potential can happen there. Yeah. So this process of, these exposed areas of the axon membrane allow for high conduction speeds by allowing the signal to hop from node to node, a phenomenon called saltatory conduction. We'll understand why it's saltatory conduction because it's all about salt. So signals are passed from neuron to neuron through the synapse, which is made of the presynaptic neuron's nerve terminal, a space called the synaptic cleft, and postsynaptic neuron membrane. So let's understand what that is with the picture. Synapse. So we were just looking at a neuron, right? We have a neuron here. So let's pretend that a signal just came in, right? And then the signal travels down the axon. This does not appear to be myelinated, but maybe they didn't have enough space to draw it. So let's just pretend that it's myelinated, right? And the signal hops like this, right? Hops down, down, down until it hits these little terminals, right? These little, so notice that over here is dendrites, right? Over here is the axon terminals, right? They're not the same thing. If it's branching off from the axon, it's an axon terminal. And once the signal hits here, right, you'll notice that on this side over here is a terminal, right? And then on this side of the next neuron is called a dendrite, right? So the signal is hopping from a terminal to a dendrite. And once enough signal comes from the terminals to the dendrite, that's going to trigger the action potential to happen in this neuron, right? Once it gets enough signal, it reaches threshold, and then we'll have the signal propagate down this axon, right? And so on and so forth. And that's how a signal gets passed on. So in each of these areas, we're going to zoom in and see like what's actually happening, right? Does that make sense so far? Hopefully, I think so far we haven't gotten into the really complicated part yet. But this is just like a gross understanding of how the signal is passing down. And so you see the terminology here, presynaptic neuron and postsynaptic neuron. Why they're saying that is because the synapse is this gap where the electrical signal has to make a jump. So you see there's a gap here, there's a gap here, there's a gap here, and there's a gap here. So there's a presynapse and a postsynapse. And that's what that's referring to. So signals are passed from neuron to neuron through the synapse, as we talked about. Over time, neurons begin to form interconnected pathways that strengthen learned knowledge. This is referred to as synaptic plasticity. So you may have heard, like, you may have seen that little kids who grow up in households speaking multiple languages can pick up on all those languages, right? But once you get older, it's harder for you to learn multiple languages, right? And they often say that kids have more plasticity in their brains. So that's what that's referring to. You have greater ability to learn when you're younger. Your brain is has more plasticity. So the plasticity part means that the parts of your brains that are being used more will strengthen, and pathways that are used less often will weaken, and pathways can even die, and that's called synaptic pruning. So what that means is, like, if we look over here, right, if this pathway is not being used that often, If this pathway doesn't get used that often, then maybe, this may just die off or be pruned, right? So that we don't have this pathway anymore. Because if we're not using it, then what's the point of keeping it around, right? Sometimes that's the case. But most neurons and active pathways remain in place for as long as possible. And neurons don't generally undergo mitosis and just remain senescent for most of their lifetime. So we talked about those. We talked about oligodendrocytes and Schwann cells. There's also some other cells, astrocytes. They provide the neurons with nutrients and they form the blood-brain barrier, right? Then there's microglia, which will phagocytose pathogens. There's ependymal cells, which are found lining the ventricles of the brain, and they produce the cerebrospinal fluid. And then those are all in the central nervous system. In the peripheral nervous system, there's satellite cells, which are just providing structural support. And as we talked about, the Schwann cells, which are for myelination. So that's that's literally some anatomy, right? Of how a neuron actually is set up. Now we need to talk about the kind of the biochemistry of how an action potential works. This is a little bit tricky. So we'll try to talk through it. And if you have any question about this, then just ask. I think I will cover this after we talk about that. Yes. So yeah, okay. Okay, so we talked before about transport, right? And we talked a little bit about the cell membrane and these kinds of things. So the membrane of the cell has a resting membrane potential, right? Has a voltage. Why? Because there are some transport proteins that are maintaining that by virtue of shuttling sodium ions, positively charged sodium ions, and positively charged potassium ions. So what's happening here is we have some leak channels, which are passive transport, which leak. So there is, just to draw this out, this is the sodium potassium pump here in purple, right? The sodium potassium pump. And what the sodium potassium pump does is it moves 3 sodium out and 2 potassium in. So 3 sodium will go out and 2 potassium will come in, right? What that's doing, because 3 positive is going out and 2 positive is coming in, that's a net of +1 to the outside and -1 to the inside, right? Does that make sense? If there's 3 positive out and 2 positive in, right, then this inside of the cell lost a positive and the outside of the cell gained a positive charge, right? Essentially. Because this is not just happening once, this is happening thousands, if not millions of times, right? This is constantly happening. And what that's eventually doing is causing a situation where the outside, the membrane is going to be polarized to -70 microvolts, -70, right? So this process happens so much that the inside of the membrane becomes -70. And this is also facilitated by these leak channels where these potassium, because this is a concentration gradient, right? There's high sodium outside, so it wants to come inside, and there's high potassium inside, so it wants to come outside, right? So the leak channels allow this, but the potassium channels are on purpose, they're more leaky. So again, we're losing more positives from the inside of the cell. So that's how we get this negative resting membrane potential. On purpose. Our cell wants to do that. Why does our cell want to have a on purpose negative resting membrane potential? Well, we're about to see. Because, so let's think back to that neuron that we were looking at, right? Let's think back to this neuron. So this neuron is chilling, and then he suddenly gets an electrical signal, right? He gets an electrical signal, and that electrical signal is actually positive charge, because it's when the electrical signal is happening, right? It is causing an influx of sodium into the cell. So when the sodium is allowed into the cell, it increases the voltage in the cell. So what's happening here is that once enough sodium comes into the cell, it triggers what's called voltage threshold or threshold potential. So how does that work? So in the resting phase, right? In the resting phase, there's no there's not, I mean, there is movement of ions, but there's no net movement of ions. There's no, it's just station, it's just no significant change, right? In either direction. Now, when the cell or when the neuron starts receiving those electrical signals and enough so then that's when sodium starts, kind of filling into the cell. Once enough sodium fills into the cell, which is at negative 55 millivolts, the threshold potential, that triggers an opening of voltage-gated sodium channels. So when the voltage-gated so you'll see here that, oh no, this is not exactly. Yeah, once the membrane voltage increases to the threshold value of around negative 50 millivolts, voltage-gated sodium channels open and allow an influx of sodium ions into one segment of the axon. So what that means, so there's a difference here. Before we hit threshold, right, there is some sodium coming in from these excited, there is some just from leak channels, but it's the excitatory electrical signals that are increasing the membrane potential a little bit. Once that membrane potential has sufficiently increased, right, that's when the floodgates essentially open. And the voltage gated sodium channels, right, they're voltage gated because they won't open until a certain voltage is hit. Once that voltage is hit, the sodium is allowed to come in. Because that sodium is allowed to come in a rush, boom, we get a huge spike. We get a huge spike in the voltage, right? Before it was negative because it was dumping all of its sodium out. Now all the sodium is allowed to come back in and it's now positive, right? It's very positive now. So on the action potential graph, this is what it looks like. At rest, we were at negative 70. Then we got some electrical signals that pushed us up a little bit. Once we hit the voltage, once we hit the threshold here, right? Right here is where those voltage-gated sodium channels open and cause this whole floodgates, right? Cause this sodium influx, right? And that's allowing us to, that's an action potential, right? Now, and so what I want you to understand is like how to link this to the anatomy, right? Because what I'm talking about is, if we look at that picture, right? If you look at this picture, right, you see the voltage gated channels. So what I'm talking about here with the sodium rushing in is happening right here. When the sodium rushes in, here, that generates an action potential. That action potential will then jump over here and more sodium will rush in. That's essentially what the action potential actually is, the sodium rushing in. So once the sodium rushes in, then Then once the sodium is rushed in and the signal has been passed on, right? Then we need to go back to normal. The reason we need to go back to the resting state is because if we're stuck up here at high membrane potential, right? Then the cell is filled with sodium, right? If the cell is already filled with sodium, it can't really effectively accept more sodium for the next action potential. So we need to go back to our resting state. So for us to go back to the resting state, it's actually going to be handled by the potassium, right? So what the potassium will do is, so the sodium channels have closed because we hit this membrane potential. And now the potassium channels will open and they will dump all of its potassium from the cell. So remember, this is also positively charged, right? So either way, we're shedding positive charge from the cell. So we're shedding. We're slowly getting back to our membrane potential. But then here at -70, something interesting happens. We actually go past -70. And why do we do that? Why do we go past -70? So, sorry, I should have mentioned the terminology. Depolarizing, right, is saying it's depolarizing when it's becoming more positive. So when sodium is entering, and then it's repolarizing when it's becoming more negative, right, going back to normal. But then we have something called hyperpolarizing. It's going more negative than it was supposed to be, right? More negative. Why is it going more negative than it was supposed to be? Well, this is actually not a bug, it's a feature. When we go a little bit more negative than we're supposed to, that prevents the neuron from immediately being triggered again. Because it's more negative, it takes it a little bit more time to get back to resting membrane potential. And once it's back at resting membrane potential, now it can accept a new signal and this action potential can happen again. So this is actually very important. The hyperpolarizing phase is actually very important because it prevents a neuron from getting like kind of like locked in continuously. And that's actually how you have like tetanus, where a neuron is just being constantly fired and has no relaxation period, no refractory period. So if we look at this, right, we can see that once we hit voltage threshold, we have the repolarization with the sodium voltage gated channels opening and the sodium influx. Then we have repolarization where the potassium channels open and allow all the potassium to leak out. Then even though we hit resting potential, we go a little bit deeper into the negative voltage, and then we come back to normal. And then this whole cycle can continue again for a neuron. So this is pretty in-depth. And so that's called the refractory period, right? Membrane potential actually becomes lower than the resting potential. This places the neuron into an absolute refractory period, during which no stimuli can trigger another action potential. The sodium-potassium pump also works to restore the resting membrane potential. Recall that for every 1 molecule of ATP that is hydrolyzed, 3 Na ions are pumped out and 2 K ions are pumped back in. The cell transitions to the relative refractory period, during which an action potential can occur only if the stimulus is greater than usual. So what that means is that the relative refractory period, so there is a point where like we won't, the neuron will not respond because it's negatively charged. It's so negative, the membrane potential is so negative. Yeah, so when this is so negative, right? When at this point, actually, wait. So absolute refractory, right, is over here, where we're already in the process of the action potential, right? The sodium is coming in or the potassium is leaving. At this point, another action potential cannot happen yet because our membrane potential is way too high, right? We need to come back down to the resting membrane potential. During this phase, right, if there's enough signaling, if there's enough positivity, right, shooting the negative, shooting this back up, right? If you put in enough electricity to shoot this back up, it will shoot back up and you will trigger another action potential, right? But so that's why this is called the relative refractory period, right? Because under normal conditions, whatever electricity you put in here, right, should not be enough, right? That's an interesting thing. The electricity that you put in for the first action potential, right, at -70, you have to put in a little bit more because you're starting at a lower threshold or memory potential. So you would need even more positive signals to push it back into an action potential. So this is how they the neuron regulates itself and doesn't get locked up doing too much, doing too many action potentials in quick succession. Okay, so this is kind of showing you, this is linking it back to the anatomy, right? So neural impulses are transmitted segmentally in the axon, right? So this is the axon, right? Once the sodium influx comes in here, it's going to encourage the next segment of the axon to also have a sodium influx. And then this will also have a sodium influx. And then this will also have a sodium influx. And the cool thing is, right, you might wonder, like, how does the signal know that it's only supposed to go down the axon? How come it never goes back up, right? Well, it's built in because while the sodium influx is happening here, right, we're having potassium efflux here, and the voltage-gated sodium channels are closed. So even if the sodium wanted to go back in here, it can't. It can't. It's already been locked out of here. So the sodium will have to go down here. And that's how we get a continuous transmission down the axon rather than backing up. Okay, so that's pretty complex. Any questions about how the role of the sodium and the ions in the action potential? Okay, then we'll talk about the neural impulses and the neurotransmitters. Okay, so we're getting to the other side of this, right? So if we look back at our neuron, right? What we have covered now is the electrical signal coming in here, then the electrical signal hopping here through these little voltage-gated sodium channels, right? Hop, 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 and then over here. So now at the synapse, right, what happens? How does the signal get passed on, right? That's a question. How does the signal get passed on? And it's actually pretty interesting. Once the electrical signal arrives at the axon terminal, there is a physical gap, right? Remember, we mentioned that there's a physical gap between the two neurons. So this here is the presynaptic neuron. This is the synapse itself, the gap, the physical gap, and then this is the postsynaptic neuron, right? So how do we bridge that gap? How do they communicate with each other? You may have heard of something called a neurotransmitter. And this is what a neurotransmitter is. It is a molecule that allows for the transmission of a signal between neurons. They are small molecule chemical messengers. So at the nerve terminal of the presynaptic neuron, the action potential triggers the influx of calcium ions by opening voltage-gated calcium channels. This causes membrane-bound vesicles to release neurotransmitters into the synaptic cleft. The neurotransmitters then bind to receptors on the membrane of the postsynaptic neuron. So what does that look like? So here's a presynaptic cell. Here's the cleft where there's just space, and then the postsynaptic cell. Once the action potential arrives, that triggers a voltage, the voltage changes in the axon terminal, voltage-gated calcium channels open, calcium comes in, and calcium actually has the ability to move these vesicles to the end of the axon terminal. What's inside these vesicles are just stored neurotransmitters. So once they get the signal from calcium, they move down to the end, right? They release the neurotransmitter. The neurotransmitter travels across the synaptic cleft and binds to the receptors on the postsynaptic neuron. And now that they have bound here, right, this is going to trigger the action potential to propagate in this new neuron. So this is the baton pass, you can think of it, right? Between two neurons, they're passing the information and they're moving forward. So the interesting thing is that the neurotransmitter itself has a big impact on what the signal is, right? It can be excitatory, which tells the neuron, hey, produce an action potential now, right? Or it can be inhibitory, and it can actually tell the postsynaptic neuron, don't produce an action potential, right? So there's a lot of different things that can be modified in this communication system, right? And we talked about those electrical signals, and we talked about, reaching threshold, right? And there's two major ways for this to happen. Temporal summation means that the one axon terminal is sending those neurotransmitters over time, like repeatedly, right? So maybe just pretend like this thing needs, like, I don't know, 20 neurotransmitters to shoot off an action potential, right? Maybe in the first vesicle, it only gets 5. And then maybe in the second vesicle, it gets 5. And then maybe in the third one, it gets 5. Fourth one, it gets 5, right? So if this one axon terminal gives 4 vesicles in quick succession, that's going to be enough neurotransmitter to trigger this action potential, right? However, what if this neuron is only releasing 1 vesicle at a time, right? In that case, it only gets 5, right? And then that's not enough. But if there are multiple axon terminals, right? If there are multiple, right? Imagine that there's like 3 more, right? Then this one gives 1, this one gives 1, and then it gets another 2 from somewhere else, right? Then it has enough. And that's called spatial summation. So it's a matter of how is the signal being added, how is the signal growing enough to trigger that action potential? And it's one of 2 ways. Either one axon is giving a bunch of vesicles, or multiple axons are sending smaller signals, right? But they're adding up. So in both ways, they're adding up to trigger that action potential. So once those neurotransmitters are released, right? If they stick around in that synaptic cleft, that could also be a problem, just in the same way that we talked about how this has to return to resting potential, right? But there's a refractory period where we don't want it to continue getting the signals, right? We also need to clear out those small chemicals from the synaptic cleft so that we're not constantly signaling this when we don't want to be signaling. So there's a couple different ways. There's a reuptake, which is that the presynaptic neuron reclaims its neurotransmitter. So in some way, this stuff will get bundled up and will go back up into the original neuron. That's one way. Another way is enzymatic degradation, where an enzyme comes in and just destroys the neurotransmitter. Well, not destroys, but catalyzes it. And then diffusion, right? The neurotransmitter just diffuses out. And so there's various different ways for that to happen. Now, neurotransmitters themselves are very important, and they have very, very different functions, right? So these are things that you do have to know. And again, this is all like memorizing and just like understanding like what it is, right? So acetylcholine, right, is usually excitatory in the it's usually excitatory in general, but always excitatory in the musculoskeletal system, and it involves motor movement. right? So if acetylcholine comes down onto a onto a muscle, it's telling that muscle to do some work, right? Norepinephrine is excitatory, causes the fight or flight response, those different responses that we saw, right? Dopamine, right? Excitatory or inhibitory, causes happiness and all these things, right? And we can see that there's also disorders that you can get if you don't have enough of a certain neurotransmitter, right? So glutamate and GABA are also very important because they are respectively, they're excitatory and they're inhibitory in our brains. And yeah, so there are various different neurotransmitters. They're all important. They all serve different roles. We talked about afferent and efferent neurons. There's also interneurons. Interneurons are in between, right? So if you would especially find them in reflexes. Interneurons connect the afferent and efferent neurons, relaying the afferent impulses up to the brain while also signaling an efferent response to the stimulus. So this is pretty interesting, right? In some cases, you need to, it's possible that like getting the, if we have a reflex, it allows us to kind of bypass the waiting for the efferent signal from the brain. What we can do is, yes, we'll receive the afferent signal, right? And the afferent signal will go up to the brain, but at the same time, through an interneuron, will signal for a motor function to occur. So we have, so yeah, hold on. Neurons. So interneurons are essentially, when you have this little neuron in between the afferent and the efferent, that's effectively what an interneuron is. So, right. The efferent, so the withdrawal reflex, the efferent neurons are able to coordinate multiple muscles so that you retract your hand quickly rather than waiting for the signals to reach the brain. This is a type of polysynaptic reflex arc as it involves at least one interneuron linking afferent and efferent neurons. Whereas a monosynaptic reflex arc is an afferent and efferent neuron with one synapse, no interneuron, right? So those are reflexes. And now we have upper motor neurons in the cerebral cortex transmitting information to the lower motor neurons in the spinal cord. Lower motor neurons then meet the skeletal muscle at neuromuscular junctions. Okay, so neuromuscular junctions are also so everything I've talked about is like the most basic version of how this works. However, in different areas, there's different ways that it works. So for example, one big example is the synapse that we just talked about, right? The synapse through this manner is how it normally works, right? Chemical signaling. However, there's also an electrical synapse, right? And the electrical synapse happens in cardiac muscle cells. Now, why would it happen in cardiac muscle cells? Because the heart is, we need very fast transmission and we need very in sync transmission. So we can see there's a lot of differences in the electrical and chemical synapse. In the chemical synapse, there is a delay as the neurotransmitters move across that synapse, right? In the electrical synapse, no such delay because they're directly linked. So on the right, we have chemical, and on the left, we have electrical. So you see on the right, you have this delay of those neurotransmitters floating across the synaptic cleft. But on the electrical chemical synapse, right, instead of a bridge to cross, right, they are directly connected through these gap junctions. So whenever there are ions floating around in the presynaptic neuron, they just go across into the postsynaptic neuron. So that allows it to have, greater continuity, basically, or greater communication. So already there's differences here, right? They use ions instead of neurotransmitters. They use gap junctions instead of ion channels. They can flow bidirectionally. They can flow up or down, right? Because this has no directionality to it, whereas this does have directionality. So there's a lot of unique stuff that you'll see. I've just given you like the general gist of things, whereas there's unique scenarios that you'll see, for example, like the end plate. So we talked about, so in muscles, right? We see that some of this is what we already talked about. The action potential comes down the axon, right? The calcium comes in, the synaptic vesicle is released, right? The acetylcholine comes out, it attaches to these receptors, right? But then what happens? Then a really different process is happening, because you'll see here, we're not attaching to another neuron, we're attaching to something called the motor end plate. So when we synapse onto the motor end plate, we're leading to a process that's going to allow for muscle contraction. So there's basically, the template, the main idea now, but there's ways that this will be modified and different. Okay, and so this is basically just talking about different reflexes that we have, and you can have certain increased, like too much or too little reflex, right? So if you have hypersensitive receptors in your muscles and you have greater stretch, right, or unable to stretch fully from hypertonia, clonus is that we talked about how those signals are coming too much. If the signals are coming too much, then you're having repeated contractions, right? And then you also have this extensor plantar response, where if you touch the bottom of the foot, they should curl down normally, but instead you touch the bottom of the foot and they extend up. It's actually really interesting if you go on YouTube and like look up these reflexes. They do it for like babies when they're born to test their reflexes, and you can see like when the typical and the abnormal responses to these tests. Okay, so that's basically a lot of dive into the nervous system. Let me see if there's anything else that we have missed. Yeah. So there's also an interesting thing where, let me see. So a little bit more in depth on neurotransmitters. So this is basically showing you how the autonomic nervous system is set up. And so remember, in the autonomic nervous system, we have parasympathetic and we have sympathetic nerves, right? Two different. And so the parasympathetic nerves from the central, imagine this is a spine here, right? We have a long preganglionic neuron. When we say preganglionic, so there's a ganglion here. Okay, so let me explain that. So this ganglion is in the middle of it's kind of like a bundle of, yeah, it's a group of neuron cell bodies. So okay, so we have white matter and gray matter, two different distinctions, right? On the spine, the white matter is on the outside and the gray matter is on the inside. The white matter refers to the axons, whereas the gray matter is the cell bodies, right? So in the CNS, in the PNS, right? When there's a group of cell bodies, it's called a ganglion. And so that ganglion is kind of how that ganglion shows you that ganglion is over here. So we have a long pre-ganglionic, before the ganglion neuron, right? And a signal goes through here, right? Once the signal hits here, it's going to cause the release of acetylcholine. Causes the release of acetylcholine. Once the acetylcholine comes out, it affects the ganglion, the cell's body here, and then that signal is going to travel down a short postganglionic membrane, and then we're going to release acetylcholine directly to whatever organ that we're trying to influence. That is how the parasympathetic nerves work. The sympathetic nerves, a little bit different, right? What's different? Well, the preganglionic neuron is not long, right? What's happening here is we are releasing acetylcholine, right? But then we release norepinephrine, not acetylcholine again, right? And specifically on the adrenal gland, right? The adrenal gland is the one that's releasing this norepinephrine. So we put acetylcholine on it, and that leads to norepinephrine and epinephrine being released. So two different ways of the autonomic nervous system being divided. We talked about this. Yeah, this is what we basically just talked about. Talked about this. Just see if there's any good pictures for action potential here. Yep, we talked about this. Oh yeah. Oh yeah. Okay. So this is a little bit. This is good. This is a little bit more in depth on the summation. So, like I was saying, right? You get this axon, this soma rather, is getting signals from all different kinds of axon terminals, right? You can see you can see that many different neurons have axon terminals coming into this one, right? So it's not just, one at a time. There's a lot of different connections or a lot of different signals that this neuron could be getting. And when we have EPSP and IPSP, EPSP is excitatory postsynaptic potential. I being inhibitory postsynaptic potential. So for example, if you got if this thing got one, if it got one excitatory and then one inhibitory, right? Imagining that they were both exactly the same, right? Then it would just cancel out. And this cell would not propagate the action potential, right? But if it got two excitatory and one inhibitory, maybe that would work, right? But then if it got one excitatory and two inhibitory, then maybe it wouldn't send anything, right? So it can get different signals and it can account for, it can like tally all those up. That's the process of summation, right? When the excitatory impulses add up enough to reach the threshold. But if you get inhibitory impulses and they outweigh the excitatory, then it'll cancel it and it will inhibit the action potential. So the neuron is getting many inputs from presynaptic neurons, excitatory or inhibitory. And if it only receives excitatory impulses, it will generate action potential. Yeah. So you can see, so this is pretty cool. This is showing you like in real time, the cells, the neurons kind of decision-making process. So here, here you can see it got one signal excitatory, then it got two signals excitatory, right? But then it got an inhibitory signal, right? And when it got that inhibitory signal, it was prevented from reaching threshold and prevented from doing an action potential, right? So in that situation, right? It's kind of a tug of war, right? It's trying to push it up, push it up, push it up, but then inhibitory is pulling it down, pulling down, pulling down, until we finally managed to reach that threshold potential. And once we did that, boom, immediately we hit the action potential. As soon as we hit it, then the floodgates are open and the action potential happens. So I kind of explained this before, where the spatial summation is the signals happening from this small signals happening from multiple terminals, whereas temporal summation is small signals happening from one axon in succession rapidly in order to reach that potential. So you can see this, these bumps, right? These excitatory bumps could be from multiple neurons, or it could be from one neuron just delivering it fast. Okay, so now we basically covered most of what there is to talk about with the nervous system. So now let's dive into a passage. So let's just read this together, I think. So we have, let me see. Okay, so demyelinating diseases cause destruction of the myelin sheath. Demyelination can be the result of an inflammatory process, viral infection, acquired metabolic derangement, or cardiovascular disruption. Diseases that can cause inflammatory demyelination of the CNS include multiple sclerosis, acute disseminated encephalomyelitis, ADEM, and acute hemorrhagic leukoencephalitis. Demyelinating diseases can also cause acute respiratory failure when the spinal cord is affected. So this is your typical intro paragraph that throws a lot of stuff at you, right? Now we get into the important part. A 70-year-old Greek female was hospitalized in the ICU due to acute respiratory failure requiring mechanical ventilation. The patient had a history of febrile disease 1 month previous to the visit, which included an acute onset of paraplegia, diplopia, progressive one-sided arm weakness, and dyspnea. A demyelinating peripheral nervous system disease was determined to be the underlying condition. An MRI of the brain revealed the presence of lesions involving the optic nerve, basal ganglia cerebellum, pons, and medulla oblongata. There was also extended involvement along the spinal cord. Further blood work revealed elevated counts of white blood cells within the cerebrospinal fluid. The patient required mechanical ventilation for 2 months. Then she was transferred to a rehabilitation center. 3 years later, she remains paraplegic. Since then, she has not suffered any other demyelination attack. Well, that's kind of sad. So let's see. Let's read these questions, right? Based on the information in the passage, which of the following is a possible effect of the patient's underlying condition? So what this is asking, right? First of all, it's asking, the patient has a condition, what could be an effect, right? So what was her condition? Her condition, underlying condition, right? A demyelinating peripheral nervous system disease. Okay, so how do we break this down? We know that her disease is demyelination, right? Or we can consider this to be a decrease in myelin, right? So before we start thinking about, a decrease in myelin, we have to think about what is myelin? What's the point of myelin anyway, right? What is the role of myelin? So again, this is the content part, right? This is the part that you have to learn, right? And so if we look at myelin, right? If we go back and look at myelin, and this is why it's so important to really challenge yourself with these practice questions, right? You have gone through this material now, but do you remember what myelin does, right? Maybe not. If you don't remember what myelin does, that's why these practice problems really encourage you to go back and understand, right? So let's go back and see what does myelin do. Okay, it insulates the axon, decreases surface area, and increases speed of action potential propagation. Okay, so let's write that in. Let's write that in. We can say myelin, when we have increased myelin, or when we have, when myelin exists, right? We increase our insulation. We decrease the depolarized surface area, and we increase the action potential propagation speed, right? This is essentially what the facts that we have learned. So now we have the content part. Now we come to the reasoning part, right? So the reasoning part is that this patient has a decreased myelin. So we have to see so we have to kind of link this to, we have to flip this all up, right? So increased myelin led to increased insulation, decreased myelin will be decreased insulation, right? And then increased depolarized surface area, and then decreased action potential propagation speed, right? So now we look at our questions, right? Our answers. A, decreased synaptic speed. B, decreased conduction speed of electrical signals. C, increased sensitivity in extremities. D, decreased rates of cognition. Which of these seems to be the most relevant? Which of these seems to line up with something that we've discussed here? A or B? Excellent, right? You guys are on it, right? We know it has to be decrease of speed, right? Excellent. So that's, again, our classic 50-50, right? We immediately delete two that are wrong, and then we are stuck with a little bit of a more nuanced choice, right? So now the thing to understand is, like, what is the difference between these two, right? So and this is, again, exactly why you need to be doing practice problems, right? Because you're like, oh, okay, wait, what does that mean, right? Okay, let's look. So, yeah. So here, right? Here we have action potential propagating down an axon, right? And this is what B is referring to: conduction speed of electrical signals. This process is called saltatory conduction. Saltatory because we use those sodium-potassium voltage-gated channels. Saltatory conduction. This is saltatory conduction. So this is conduction of the signal down an axon. And the other part, right, the synapse, right? The synapse speed is basically how long for these neurotransmitters to travel across the synaptic cleft. And the reason that we brought it up is because we saw that in the two different types, electrical versus chemical, the electrical one is much faster than the chemical synapse. So now knowing that, right, Which of those is related to the patient's underlying condition, right? What was the patient's underlying condition? That they had decreased myelin, right? So where did myelin play a role? In the synapse or in the conduction? Right, in the conduction, right? If we go back and look at the neuron, go back and look at the what was it called? Myelin. If we go back and look here, right? This is the myelin sheath that's allowing for this hopping, for this jumping of the signal. So good. That's it. I think this is a great question, right? I think this is an excellent question because it takes it takes a small element from the passage. Everything else, you build on, you have to know the content of what decrease of myelin does, right? And then you have to do that reasoning. No, you have to know what myelin does, right? That's the content. Then you have to do that reasoning. Okay, I know that myelin does this, so a decrease of myelin would do this. That's the reasoning. And now that you know the decrease of myelin does this, then you have your content, right? Of synaptic speed versus conduction speed. This is an excellent question. And this is if you're learning, this is a great question, right? Because look at how much you learn from this question. You force yourself to encounter, myelin's function, and then you force yourself to understand between synaptic and conductive speed. So you learn a lot from this question. I really like this question. And hopefully, once you're through this and you understand it, then you're like, oh, okay, I've learned a lot. This makes sense. Okay, cool. So let me get rid of this. Hopefully that makes sense. But if not, just post a question. And now we have question number 2. The structures that receive damage in this patient are most directly responsible for which of the following functions, right? So first of all, what was the structure that received damage in this patient? Right? What was the structure that received damage in this patient? The myelin, right? Exactly. The demyelinating peripheral nervous system was determined to be the underlying condition. And yeah. So once the myelin is impacted, right? Which function was messed up, right? Synthesizing protein and housing genetic material are just distractors, right? We have sending signals and receiving signals, right? So which of those is more relevant here, right? So the myelin, exactly, on sending, right? What is in charge of receiving? See, this is again, right, for you to really get the most out of your questions, right? Okay, maybe I know that myelin is on an axon and that sends signals. Okay, I got that. What receives signals? Challenge yourself. Don't be happy with, oh, okay, I got the answer, right? Challenge yourself. What receives signals? Excellent, exactly. Dendrites receive the signals, right? Whereas axons are myelinated and send the signals, right? And that just doing that process tells you, oh, interesting. Dendrites are not myelinated. Boom, you've learned something else, right? That's how you learn. That's how you go through the process. So good. You not only do you understand why A is correct, but you understand why B is incorrect. That's exactly what you want to be doing. Awesome. So question 3. The patient's arm weakness is most likely due to a defect in which of the following structures, right? So I believe they said that this person has left-sided, oh no, they just said one-sided arm weakness. They didn't say anything. Okay, so this person is unable to, they have muscle weakness in one of their arms, right? So the arm weakness is due to a defect in which of the following structures? So this is asking about afferent, efferent, sensory, and interneurons. Which of those do we talk about being related to the muscles? If afferent, so let's remember our mnemonic. Afferent signals arrive, efferent signals exit. Yes, correct. Efferent signals, not exist, exit, exit out of the CNS. Right? Essentially, that's what it is, right? So the efferent signals are so most likely in this patient, right? She has weakness. She doesn't have complete paralysis or anything, right? So she's able to signal somewhat, right? But so it's not her CNS that's impacted, but once those signals get there, right? Maybe there's less efferent neurons or something, right? It's most likely the efferent neurons that are impacted there, right? Because the full signal isn't getting through to the muscles. So good. Yeah, this should be efferent neurons. Now, according to the information presented in the passage, damage to which of the following structures would result in the physical symptoms presented by this patient? So what do we think? Damage to which of the following structures would result in the physical symptoms presented by this patient? So what was their damage, right? As we just said, their damage was the demyelination, right? So, and they have weakness in their efferent neurons, right? Right? So where do you think how do I phrase this? There's clearly two categories of questions here, right? There's this and there's this. So do you think it's the motor neurons or do you think it's these cells? Do you remember what these Schwann cells and oligodendrocytes do? The Schwann cells and the oligodendrocytes are these what are called glial cells. And oh, sorry, no. The glial cells are just the Schwann cells, but these guys are. Where's my picture? I had a good picture. This is just showing all of them. I showed you guys a picture before. Where is it? Here it is. So these cells are the cells that actually produce the myelin, right? So I kind of gave it away, but basically, right, this person had damage to their myelin, therefore, one of these myelin producing cells are probably impacted, right? However, the question now is to go a step further, is it the cells? So then you have to know what's the difference between the Schwann cells and the oligodendrocytes. Do you remember what the difference is between Schwann and oligodendrocyte? And again, if you don't know, if you don't remember, that's okay, right? Because this is the learning process, right? Obviously, you're not gonna remember everything that I said. That's the whole point, right? Good. So the oligodendrocytes is in the CNS and the Schwann is in the PNS, right? The way that I like to remember it is that oligodendrocyte is like a complicated name and the brain is complicated, right? So that's how I remember it. And then the Schwann cells are like are a simple name and they're not in the brain. That's what makes sense to me. But so for this patient, right, is her damage to her myelin in her central nervous system or in her peripheral nervous system? What was the symptoms that she had? She had arm weakness, right? Muscle weakness. Is that central or is that peripheral? It's peripheral. Yes, it's peripheral, right? So because it's peripheral, is it a Schwann cell or is it oligodendrocytes that are implicated? Exactly. So that's the answer, right? So we kind of broke it down a step at a time. First, we said, okay, it's myelin that's affected. So it's one of these. And then is it where is the myelin that's being affected? Well, the patient has arm weakness, right? They can send signals from the CNS, but once those signals get there, they're not responding as with full power as they should be, right? So one possibility is that the myelin that was impacted because of a deficit of Schwann cells. Okay, good. So now, question 5. Where do the upper motor neurons and lower neurons apply? Oh, so that was where I think we were talking over here. So motor neurons can be further classified into two main categories, upper motor neurons and lower motor neurons. In a nutshell, upper motor neurons in the cerebral cortex transmit information to the lower motor neurons in the spinal cord. Lower motor neurons then meet the skeletal muscle at the neuromuscular junctions. So this is going to be more in depth when we talk about muscle contraction and musculoskeletal system, because I kind of hinted at it, but we didn't go in depth into uh, we didn't go in depth into not this. What is it called? Oh, motor end plate. Yeah, we didn't go in depth into the neuromuscular junction because this is a little bit more of a separate topic. But that's basically where it applies. It applies to musculoskeletal and muscle contraction and whatnot. No problem. So where were we? Question 5. Which of the following accurately describes a relative refractory period after an action potential? Okay. Oh, and also notice, this is a very typical, like, has absolutely nothing to do with the passage, is just a slightly relevant, hey, do you know this thing, right? Which of the following accurately describes a relative refractory period after an action potential? Okay. So in the relative refractory period, we were talking about, let me just pull up the graph, relative refractory, right? This is what we were talking about here, right? The fact that after we go back down, we have this period where we hyperpolarize, right? And we go lower than our membrane potential. So which of the following accurately describes a relative refractory period after an action potential? Okay. An action potential is unable to be generated again by any stimulus. An action potential is able to be generated again by a weaker stimulus. An action potential is able to be generated again by a stronger than usual stimulus. An action potential is able to be generated again by a normal stimulus, but it becomes more energetically costly. So first of all, we have to make a decision, right? Before we get into the details, right? We have a major decision. Is it unable or is it able, right? So in the relative refractory period, is an action potential able or unable to be generated, right? And so the way that you would answer this is based on the fact that if there's a relative refractory period, then there's also an absolute refractory period, right? So the absolute refractory period is where we can't generate another action potential. Why? Because we're already in the middle of an action potential, right? Kind of common sense, right? If we're already stuck in the action potential process, it's not the time for another action potential to happen here. Because then it would be crazy. Then it would look like it would look like this, right? And then if you did it again, it would look like that. Like that's the cell doesn't do that, right? So if there's an absolute refractory period, if there's a relative refractory period, then there's also an absolute refractory period. And the absolute refractory period is when it's saying that action potential is unable to be generated by any stimulus. So this is referring to absolute refractory, not relative, right? So now when we come to relative, we have to determine it is being able to generate, but under what conditions, right? This is something that we have to piece together now. It's either by a weaker stimulus or by a stronger than usual stimulus or by a normal stimulus that's more costly, right? Okay, so we have three pretty common sense like options: weaker, stronger, or normal, right? So if you look at this graph here, right? If a signal wants to come in, if a signal wants to come in at this point, right? Right? Does the signal need to be weaker, stronger, or the same for us to go reach threshold potential? Right, stronger, right? Because we have a greater deficit, a greater amount of membrane potential to cover, right? From here, it was only like, if this is 50, as it typically is, right? From here, it's only a difference of 20, right? So it's only, we only need electrical stimulation of about 20 microvolts, right? But when we're hyperpolarized, right? Whatever this amount is, maybe it's 5 microvolts. Now we need it to cover a difference of 25, right? So that stimulation of 25 microvolts is stronger than the stimulation of 20 microvolts, right? So that leads us to an action potential is able to be generated again. However, it needs to be through the use of, it needs to be through a stronger than usual stimulus. Okay, so let's just check our answers. We said it was decreased conduction speed. We said it was sending signals because receiving is dendrites. We said it was efferent neurons. We said it was Schwann cells. And we said stronger than usual stimulus. So we had B. Oh, I forgot to take the choices. Yeah, B, B, A, B, A, B, A, C. B, A, B, A, C. Yep. Boom. So that's in a nutshell the nervous system. And yeah, so we have the next week we're going to cover the endocrine system, which is actually still part of this, but it's all part of the same content category, but it's too much to cover both in one day because the nervous system itself is quite in depth and yeah. So after this, or whenever you get a chance, right? Do your Anki cards, memorize those anatomical things, and do some practice problems, and you should be good to go on this one. Okay.